The subject matter of this video, as discussed by Lydia Middleton, Madison Pui, Elise Sims, and Brianna Sobel, will include how reproductive parameters are impacted by physiological stress and by particles found in soft plastics known as phthalates. This video will include the specifics of phthalate and stress-induced reductions of male fertility through the loss of sperm viability and production.
Decreased sperm functionality is the largest cause of infertility in humans. Such dysfunctions could be caused by low motility, low sperm count in the semen, or DNA damage originating in spermatogenesis. Motility is a measure of the quantity of sperm in the male semen that are able to swim forward towards the female egg. Low motility indicates a disability in the sperm approaching the egg, which will in turn affect the success of fertilization. Two common contributors to sperm damage and/or dysfunction are exposure to man-made chemicals and stress. Man-made soft plastics contain substances called phthalates, which show correlation with lower motility of sperm in men and subsequent difficulties with conception. Stress has been shown to decrease sperm quality in terms of functionality, quantity, and ability to fertilize. Stress, quantified in the forms of both stressful life events and the relative feeling of stress in daily life, can impact sperm viability and eventual reproductive success.
In the study done at Lund University in Sweden, samples of blood, urine, and semen from young adult males were examined to determine associations between exposure to phthalates and male fertility. The degree of exposure to phthalates was determined by measuring the presence of metabolites of specific phthalates in blood and urine. Reproductive function was quantified by looking at progressive motility, morphology, sperm count, and concentration of human semen. This table compares metabolite markers of the phthalates in urine to the reproductive characteristics of semen. We can see that the DHP metabolites, MEHP and MECPP, are negatively associated with progressive motility of semen. We can also see that all DEHP metabolites were positively associated with high DNA stainability. This table compares the degree of presence of metabolite markers in urine to the presence of reproductive hormones. Ultimately, there was no difference in reproductive hormones between men of high exposure and of low exposure for any of the phthalate metabolite markers. However, we can still see a negative association between the metabolites of DINP phthalates and the presence of follicle-stimulating hormone. This data also illustrates a negative association between percent MEHP and free testosterone.
Overall, higher levels of DEHP are negatively associated with sperm motility. Other studies about phthalate exposure have had contradictory results, but DEHP is consistently associated with lower semen quality across all studies. This is possibly due to the fact that it is the most toxic phthalate based on the negative effects that it has on the male reproductive system. A potential biological mechanism associated with this stems from disturbed acquisition of motility in the epididymis. This can result from oxidative stress, which is commonly caused by DEHP. A clinical consequence of high exposure to this toxic phthalate is a reduction in fertility due to the fact that sperm motility is linearly related to pregnancy rates. A main point of this study in particular is that phthalate exposure during adulthood, instead only during the sensitive period of fetal development, can negatively affect reproduction. Also, this study was the first of its kind to take into consideration the urine concentration as well as the length of time since the last ejaculation. And most of all, the men that were studied were from the general population instead of from a sample of males with pre-existing fertility problems that could cause potentially extraneous variables in the analysis of sperm quality. DEHP is considered a hazardous substance by the European Union and is not allowed to be used in production of products, but it does not account for imported goods. However, there is no intense need for immediate concern since the substances will usually break down in the body after a couple of days.
It's been discovered that a man's social environment serves as a factor in determining his reproductive health. Both subjective and objective indicators of stress should be reviewed in observing associated sperm appearance, motility, and concentration. Also, research through the years has found that there is a linear correlation between sperm motility and fertility, and the findings herein could also predict the relationship between DEHP exposure and fertility and its consequences. Additionally, work that increases an understanding of the genetic aspects of fertility as it relates to in vitro fertilization may allow researchers to determine exactly which proteins are required to build on the efficiency of the IVF process and ultimately improve the health of the infant.
The information in this video was obtained from the following articles and sources. Columbia University's study provided results on stress's harmful effects on sperm and semen quality, while Lund University's study covered the impact of DEHP, a known phthalate, on sperm. The first study by Axelsen provided further data on DEHP metabolite levels and observed associations with reproductive parameters, and the second also observed the DEHP levels and included DINP levels in conjunction with hormone levels and semen volume. The article obtained from the EBSCO database furthered existing information taken from the other studies, and the Atkin article covered how DNA damage reduces fertility, giving context as to possible direct effects of phthalates and how they could possibly reduce sperm viability and functionality.